Hi there, welcome to DNA lecture video number 5. This is our last video for this unit. Let's begin. Let's do a quick review. This is where we ended last time. We spoke last time. These are the two processes, transcription and translation, are the two processes involved in protein synthesis. Transcription, we talked about last time. This is the process in which DNA, is then converted into RNA. In any kind of RNA, all right? So DNA gets turned into RNA, that is transcription, all right? Then this RNA eventually can get turned into a protein. In this case, it's called a polypeptide. Polypeptide just hasn't folded and become a protein yet, but polypeptides become proteins. So for what you need to know, you can assume this is a protein. So today we're going to discuss translation, the part that takes the RNA and converts it into a protein, or in this case, a polypeptide. All right, let's begin. So let's talk about the genetic code. Now, the genetic code is the letters in your code, the A's, C's, T's, and G's in DNA. Okay, the A's, C's, T's, and G's, once again, in DNA can get converted into RNA using the complementary base pairing rules. RNA kind of holds the code. Messenger RNA kind of holds the code. This is called a codon chart. These words in this chart, phenylalanine, leucine, serine, tyrosine, cysteine, tryptophan, histidine, glutamine, proline, isoleucine, methionine, threonine, alanine, valine, aspartic acid, glutamic acid, glycine, arginine, all these words here, these are the names of the amino acids. These are the 20 different building blocks of DNA. You'll probably notice there's a stop codon, and I'll explain to you what that means in a second. Methionine over here, methionine, just so you know, is the start codon. All right. So methionine, yes, it is an amino acid. It is also the amino acid that pretty much starts the beginnings of every protein. All right. You don't need to know this for your test, but proteins get modified later on after they're produced, and sometimes the ends might get cut off. So methionine will begin, it'll be in the polypeptide or the protein always at the beginning. It starts every polypeptide or protein. Okay. It begins every one of them, but it could get cut off when that protein gets processed. So it may not end up being in the functioning protein at the end. Okay. So methionine is the start or the first amino acid in every protein sequence that you're going to need to deal with. And eventually it ends with what's called a stop signal. There's 3 stop signals. Now you have codons. What's this word mean? 3 messenger RNA bases. All right. So for example, if you have AUG, and this is messenger RNA. Okay. This is not DNA. There's no T here. So DNA got converted in this. What DNA code got turned in this? Well, DNA was T became A, U became TAC. T became A became U, C became G. So AUG is this is a codon. This is messenger RNA. This is not DNA. You look up AUG, the first letter, first base, A. The second base, U. So A is here somewhere in this row. U is in this column. So now we've narrowed it down to this box here. The G is over here. So A, U, we're in this box. G puts us at methionine. Methionine is the start codon. It is the amino acid that starts every protein sequence that you will be dealing with for our class. Okay. There's 3 stop signals or stop codons. UAG is the first one. So the first letter is U. Second letter is A. That puts us in this box here. Third letter is G right there. UAG. The second one, UGA. UGA. We're in this box right here. Third one, UAA. UAA. We're right there. Stop. Okay, so this is how you read these charts. Yes, you will be given a chart to read. You will need to be able to understand the chart and read the chart. So what are the words in here again to remind you? These are the names of the amino acids. These are the building blocks of protein. You connect them in chains. Those chains then fold on each other and create shapes and create functioning proteins. Anticodon is the opposite of the codon. I'm going to explain that to you in a little bit using a picture. A lot of students mess up the anticodons in the test. So when I explain it to you shortly, I would pay attention to what exactly an anticodon is. I see a lot of students struggling on these questions on the test. Okay. The genetic code, just so you know, this code it's almost universal, which means all living things on the planet pretty much use the same code to produce their proteins. So remember, DNA gets converted into messenger RNA. 3 messenger RNA bases, so 3 letters in messenger RNA, make up a codon, and this is a codon chart. So this chart specifically requires you to use mRNA coding, not DNA. DNA needs to get converted into messenger RNA. I'm going to show you how to do all that. Protein structure. Let's talk about proteins. Proteins are made out of amino acids. So those things I just talked about, that codon chart full of amino acids, phenylalanine, isoleucine, asparagine, all those names, those are all the different amino acids that you link together to produce proteins. There's 20 different amino acids. Polypeptide, I've already generally said that. That is a chain of amino acids. That's a polypeptide. Peptide bond, that's the bond that holds amino acids to each other, next to each other. Okay, that's what a peptide bond is. Proteins, they're not always made out of one chain. The definition of a protein, a protein is a polypeptide with a function. Okay, and if it doesn't have a function, then it's not a protein. It's just a polypeptide. Okay, so proteins can be made out of one polypeptide. They can be made out of multiple polypeptides connected to each other. So proteins are not always made up of one chain of amino acids. Okay, that's a concept that a lot of students don't understand. In a chain of amino acids or in a polypeptide, you can have hundreds or thousands. To keep it simple, the ones we're going to do in class, the examples, the samples you're going to have, they're going to have, I don't know, 3, 4, 5, 6 of them in the chain. I'm not going to have more than that. All right. So the amino acids determines how the polypeptide twists around itself, and the 3-dimensional structure that this talks about. Now that it's 3-dimensional, it's not just a chain. Now it has a shape. Now it'll have a function. And once it has a function, now it has a job. Now it's called a protein. This is showing you a picture of a blood protein called hemoglobin. It's hard to see, but the iron's right there. Fe's iron. This is a blood protein that carries oxygen. It's made out of these 4 different chains of amino acids all combined together. So 4 different polypeptides folded up, made a 3-dimensional structure that can carry oxygen. That is a protein right there. It has a function. Now, so we talked about transcription in our last video. Now we got to talk about translation in this video. Okay, I'm going to talk about anticodons in a second, so I would pay attention so you have an understanding of what an anticodon is. So what is translation? Translation, what are we translating? You're taking the code, which was turned into RNA, messenger RNA, and you're taking that mRNA, that code in the messenger RNA, and it is being read by a ribosome, and the ribosome is this brownish tan area on the bottom here and up top here. It's being read by the ribosome, and it gets converted into a protein, or technically a polypeptide first, and then that becomes a protein. What is involved in translation? Well, all of the RNA that you learned about is involved in translation. mRNA, okay, that is this pink thing right here. The pink is mRNA, all right? tRNA is the green thing. This is tRNA, okay? And rRNA, ribosomal RNA, it's not labeled, but that's the brownish tan stuff. That's the ribosome. That is what the ribosome is made out of. So all of the RNA is involved in translation. And translation, once again, turns mRNA, the pink thing, into a protein made out of chains of these guys called amino acids. Where does it happen? Well, it happens in the ribosomes, and they're in the cytoplasm in your cells, eukaryotes. Bacteria, they don't have a nucleus, so it always happens in their cytoplasm. That's the only place they have material stored. They don't have anything in their nucleus. So where does translation take place? It always takes place in the ribosomes, in the cytoplasm. Okay? Step 1. All right, let's talk about the steps. And in the steps, I'm going to explain to you some details. All right? Step 1 of translation. the ribosome comes together. So you have a small subunit and a large subunit. They come together. Okay, so you small one and a large one, they clamp together and they connect over the messenger RNA. So the messenger RNA gets fed through the ribosome. It goes right through the ribosome, and that's how the ribosome reads it. Okay, on one side, now pay attention. You have the pink thing is messenger RNA. Messenger RNA has codons. Every 3 letters, 3 bases on messenger RNA is called a codon. All right. The 3 opposite letters or bases on the tRNA are called anticodons. And I'm going to explain to you, showing you a picture in a second so you can go in more detail. How does the tRNA know where to land? The codon from see the pink? That's the codon. The codon matches the opposite anticodon. So A goes to U goes to A, G goes to C. Once the tRNA knows how it matches up, now it knows to lay down the amino acid. And this is methionine. This is the first amino acid that always gets laid down because it is the start signal. So it always begins the sequence. So this is the start codon, AUG. AUG is methionine. How does the tRNA know where to land? Its opposite anticodon balances and matches up to the codon. Okay, so that's how this works. Here's another picture showing it to you a little bit more detail. All right, let me move this up for a second. So here's what we got here. So this is the start codon, AUG. So this is a codon chart. So let's look up AUG. AUG. AU puts us in this one, and G is the bottom here, is methionine. You see how that says MET? That's methionine. Okay, so this is the start. AUG is a codon. This is a codon chart. This is not a tRNA chart. This is not an anticodon chart. This is a codon chart. You have to have it as mRNA. Now, if I ask you for the anticodon of methionine, I want the opposite of it, the complementary RNA bases to it. So the start codon is AUG. The start anticodon is UAC. The opposite of A is U. The opposite of U is A. The opposite of G is C. So on the test, if I ask you about anticodons, you need to convert codons into anticodons using the base pairing rules. All right, bring the picture back down. Here we go. Now, step 2. All right, so step 1 is the start signal. Step 2 is this mRNA gets fed through the ribosome. And as it's fed through the ribosome, tRNA, these green things, they keep matching up their anticodons to the codons. Anticodon matches up codon, and then they keep dropping off amino acids, creating a chain called a polypeptide. Okay, so it keeps dropping off amino acids as a result of anticodons matching up to codons. That's how it knows where to lay down the amino acids. Step 3, the chain keeps growing, and eventually there's a stop signal called the stop codon. Okay, when it gets to the stop codon, the polypeptide gets released. Okay, termination happens. It's done. All right, it's reached the end of the line. All right, as this occurs, this codon, this ribosome can come apart, or it can continue translating if it needs to, or it can come apart and reassemble later if needed. All right, so the whole idea is mRNA gets fed through a ribosome. The tRNA's anticodons match up to the codons. It keeps laying down amino acids. That's these things over here. It creates a chain of amino acids called a polypeptide, and eventually it hits a stop signal or the stop codon, and then the polypeptide gets released, and now you have your protein. Okay, all right, and let's review how they're related to each other. Transcription takes the DNA, turns it into RNA. Okay, this RNA, if it's messenger RNA gets fed through a ribosome. And as it gets fed through a ribosome, the 3 anticodons on the tRNA match up to the 3 codons on the DNA. So look here, AAC, that's an anticodon because that's tRNA. What's the opposite of it? The codon is UUG. Okay. Over here, the anticodon of this one is CUU. What's the codon? GAA. So codons are mRNA, anticodons are tRNA. These are your tRNAs here carrying these little black things called amino acids. It's creating a chain of amino acids, creating a polypeptide that will become a protein. Right? Now, these pictures will help explain this next part. Now, when you make, this is a eukaryotic cell, this could be you or me. We make mRNA, and that's mRNA is this green thing over here, right? Getting released, all right? The mRNA gets fed through the ribosomes, all right? You and our cells, we could have one mRNA transcript, is what we call it. We can have one mRNA transcript being read by multiple ribosomes at the same time. Why is this a good thing? Then our cells can make proteins more efficiently. So you only have one mRNA, but you have multiple ribosomes, so you're producing more protein. You're producing more protein that way. So that way it's helpful to us. We can be more efficient. All right. Bacteria, because they don't have a nucleus, and by the way, let me go back a second. Eukaryotes, they have to do transcription separate than translation because transcription is in the nucleus. All right, so the DNA is stuck in the nucleus, and then the RNA has to leave. So it basically finishes transcription, then it begins translation. Bacteria are a little different. There's no nucleus. So they can make the mRNA, and that mRNA can start being read by ribosomes before the mRNA is even finished being made. So bacteria, prokaryotes, can begin making, they can do translation at the same time they're doing transcription. So the mRNA that's done starts getting fed through the ribosomes right here, and these ribosomes start making their proteins while the bacteria, the DNA, is still getting converted into mRNA through transcription. So bacteria can do transcription and translation simultaneously, which helps make them even, a little bit more efficient. Let's talk about the human genome in general, and I've mentioned this in previous chapters. We've got the coding for the entire human genome. So all of your DNA has been sequenced. The company here in San Diego, they're called Illumina. As I'm making this video, I believe it cost about $10,000 if you want to have your DNA sequence. You can actually have your DNA sequence if you want. I believe that's the going rate right now. It'll eventually be cheap, I'm assuming. You have 46 chromosomes, 23 pairs. You have 3.2 billion base pairs. That's a lot of pairs of DNA. Bioinformatics, what you're going to start seeing in the future of science is computers doing simulations, basically, and what kind of proteins are you going to make based off of this DNA sequence. So that's called bioinformatics, using computers and programming to help figure out what's happening biologically. You can do a PhD in bioinformatics and you become a doctor in this particular field of biology, and you can be someone that people hire to help simulate what a protein will look like based off of a certain sequence. And it's kind of the future of science. We have about 30,000 genes in our cells. Okay, it just shows you why is knowing the sequencing of a human of our genes and our human DNA. Why is it a good thing? Well, it's going to help us down the road with treating diseases and treating ailments and things of that sort. So it's going to be huge in medicine. All right. So now we have a protein synthesis sample. Okay. I'm going to do it in slides. I'm going to show you. You should write down what you see on each time I click to the next slide. So here's your sample. Okay, you have a sequence of DNA, and not all of the sequence of DNA is important. A lot of your DNA is not important, meaning it doesn't do anything. It kind of just protects the other DNA. It's just there. So if a mutation happens in the part of your DNA, we call it the non-coding DNA. And most of your DNA, 99% of your DNA is non-coding DNA. A lot of your DNA that's non-coding DNA, that DNA kind of protects the other DNA. So is there a purpose for it? I guess it protects it. There's probably purposes that we have not discovered yet in that non-coding DNA. All right. So ideally, for what we're dealing with our class, we're keeping this simple. All we really want to deal with is the coding part of the DNA. That's the gene. So step one says locate the gene. And this says the gene is found between the promoter. It says promote. Let's just say promoter sequence and the terminal sequence. Okay. How do we know where the promoter sequence is? Well, you look for AC. I give it to you. This is given. It's ACTA. So you go along. You're like, oh, look, ACTA. That's a promoter. Okay. So that's where it begins. That's not quite the gene yet. The gene is between start and stop, but the gene happens right after the promoter. So ACTA is the promoter. That means the gene is going to start right after it. Okay. GGAT is over here. GGAT, that's the terminal. So the gene is found between the promoter sequence and the terminal sequence. So here's where they're at. There's the promoter. There's the terminal sequence. So I've done it now I've located the gene for you. Maybe you should put this in parentheses on your paper so that you know that you found the promoter sequence, okay? And you know that you found the terminal sequence. Right here, okay? So the gene is everything between there. That's the important part. So TAC all the way over here to ATC, okay? So step one, locate it. So you find the promoter, you find the terminal sequence. I would put brackets around them. I made mine red so that you could see them. The gene is what's in between there. Okay. The next step two, transcribe the gene. Okay. What does that mean? You rewrite the DNA code into an mRNA code according to the base pairing rules. So what does T become? T becomes A. A becomes U. C becomes G. T becomes A, T becomes A, C becomes G, A becomes U, G becomes C, A becomes U, T becomes A, and so on and so forth. You just do the gene. That's the area between the promoter and between the terminal sequence. Here's what your paper should look like after you've transcribed it. Okay? I color-coded it. I even spread it out a little bit. What I would do on your paper, I would put a line between each three bases so that they don't overlap and create confusion for you. I have color-coded it and put spaces between them to show you how it's done. Okay, so we have our promoter sequence, we have our terminal sequence. What's between there? This is our gene. Our gene is between TAC and all the way to ATC. This is our gene. And they are read in groups of three. They're called codons. AUG is a codon, AAG is a codon, UCU is a codon, AGU is a codon, AUC is a codon, and UAG is a codon. Okay, so this is the important part here. All right, so now we located it, step one. Step two, we've now transcribed it. We've converted the gene into mRNA by using the complementary base pairing rules. Okay, the final step three, translate it, which means you take this mRNA sequence and you convert it into amino acids. And those amino acids will form the protein. And what does it say? Use the chart. So let me show you what this next step looks like. Okay, here's what the chart looks like. I'm going to put my picture up here. All right, here's what the chart looks like. So let's begin. Let's show you how it's done. And by the way, everything before start and after, you don't need it. There's no value to it. It's non-coding DNA. We don't need that DNA. That DNA is not involved in the actual protein that we will see. So your protein, this is important. Your protein is always start to stop. So look at the protein is the amino acid sequence from start to the stop signal. So let's first do it. AUG, let's look it up. AU, that puts us here. G, that's methionine. That's the start signal. So AUG is methionine. So write down methionine on your paper. That corresponds with AUG. The next one is AAG. A, that puts us in this one, G. The next one is lysine. So I wrote down lysine. AAG corresponds to lysine. UCU is the next one. UC, third letter U, serine. So UCU is serine. That goes right there. AGU, AGU, serine again. So yes, words can be duplicated. It's okay. They're building blocks. It just means you needed the same building block twice. Okay. So AUG is methionine. AAG is lysine. UCU is serine. AGU is also serine. The next one, AUC. AUC. Isoleucine is next. We have isoleucine there. AUC is isoleucine. And UAG. UAG is the stop signal. Stop. Okay. Sometimes you may, on the test, the word stop may not be there as the choice. If it's there, that's fine, pick it. But stop is not necessarily included in the protein. Why? Stop it's not an amino acid. It's a signal that says you are done. Okay, so the protein is always between the start and the stop. Everything after, non-coding. Everything before, non-coding DNA. So make sure if I give you a sequence of DNA, you convert it into messenger RNA, and your protein is the start to the stop signal. Everything between there. Okay, that is what produces the protein. All right, we will end our lecture there.